Welcome, everyone. It's the top of the hour. I'm Dia Waguespack in Houston, and I would like to welcome you to today's GlomCom seminar. Today, our talk is titled APOL1 Kidney Disease, Genetic Basis and Its Modifications. And we are very excited to have our speaker, Dr. Simone Sanna-Cherchi.
Thank you, Dia, for the kind introduction. Thank you, everybody, for being here. It's a very large audience. That's exciting. So let me get to the presentation. And it's a real pleasure to be surrounded by many people in these tough times. I come from Columbia University. You've seen the news, so show me some love. All right, so today I have a pretty packed presentation. We can skip some stuff, but feel free to interrupt me with questions or save them for the end. So I'll go straight to the juice. I don't have personal financial disclosure. The only thing I can say is that AstraZeneca is supporting some of our sequencing effort for nephrotic syndrome, especially, and for FSGS. So today we're going to talk about the ancestry-driven risk for kidney disease, right? So we know approximately that individuals of genetic African ancestry, they have a twofold increased risk of ESKD, 3 to 10-fold increased risk of FSGS, and 18 to 100-fold increased risk of HIV-associated nephropathy, which is basically a form of FSGS, collapsing FSGS. So the genetic support to this ancestry-driven risk disparity comes from two very, very elegant studies dating back in 2008 from Jeffrey Kopp and Linda Cao, studying non-diabetic kidney disease and FSGS, in which they basically apply a mixture mapping to population cohorts from the United States that are mixed. Right? So in a situation in which a genetic admixture exists and there is a susceptibility locus that is coming from one of the two genetic ancestries, the idea is that at this locus there will be an excess of local genetic ancestry for the population that has the higher risk, or a decrease of genetic ancestry for the population that is, let's say, protected. So by doing this study, they show that there is, as expected in individual of African ancestry in the United States, there is a general 80-20% of genetic ancestry from Africa and Europe, respectively, except that the chromosome 22 locus. Here, there was a dramatically and significantly increased ancestry, local ancestry for genome from Africa, and they mapped it to these regions on chromosome 22, around 35 megabases, where initially they attributed the risk to MYH9 being the most obvious candidate. A couple of years afterwards, two studies, the one from Genovese in Science from Martin Borlaug Group and the one in Human Genetics from Zur from Skorecki, they actually figure out that this ancestry-driven risk was not driven by variants in MYH9, but by two mutually exclusive variants named G1 and G2 on APOL1. So APOL1 kidney disease is pretty exciting and is very unique, is exemplary of one of the few examples of common variants with a very large effect size. In fact, if you look at this review from Terry Manol, you're now already back to 2009, this is sort of like a spectrum of allelic frequency and effect size for any given trait, right? So if you look at the x-axis, here we have the allelic frequency for the given gene from very rare to very common, and on the y-axis the effect size, so the penetrance of the odds ratio, right? So the expectation is that rare disease is caused by Mendelian disease. They will have rare variants, ultra rare, that they have very, very large effect size, so where the genetic correlation between the gene and phenotype is very high. Then you go down in this axis towards common, and the more they become common, the more the effect size decreases, and you get out of this extreme where common variants usually are the one that they constitute this so-called polygenic background that predisposes to complex traits and more common and frequent diseases. There are rare variants with small effect size that are probably less relevant, or just we are not able to really capture. And there are very few examples of variants that are very common, but yet they have a very high effect size, and this is APOL1, right? So why these unlikely genetic scenarios they can happen, and we'll see it very soon. So what do we know about APOL1? So before 2010, we didn't know much. We just, at least from the kidney perspective. So we knew that is an immune innate factor that circulates and is bound to high density lipoprotein. Nothing was known about the kidney. So there are two independent and mutually exclusive risk alleles, this G1 and G2. The G1 are two missense alleles that are in tight linkage disequilibrium, meaning that they're traveling nearly always together. And then there's G2 that is a six base pair deletion. They are in the same exon, the terminal part of APOL1. And G0 is considered the reference allele. So the risk for kidney disease coming from a recessive model. So high-risk genotype are the homozygous for either G1 or G2, or the compounded heterozygous G1, G2. All the others are considered non-risk or low-risk alleles. So if you look at population-based studies, the frequency of having two risk alleles is pretty high in West African, and I'll show you something later that is more precise, but up to 1/3 or 1/4 of individuals in West Africa, they have two risk alleles. Individuals self-reported Black from the United States, 13%, they have a high-risk genotype, and in individuals self-reported Hispanic or Latinx genetically, about 1% to 2.1%, they have a high-risk allele. So why are they so common if they impart so much risk for kidney disease? And the reason stands in this beautiful story of evolution and the perpetual fight between humans and the human DNA and the infections, right? So if you ask a geneticist what causes any disease, will say genetics. And if you ask an infectious disease doctor what causes disease, an infectious disease doctor will tell you everything is infectious. In reality, this perpetual battle. So we evolve our genome in order to fight infection or aggression from outside. So in this case,